Remember the times before 2020? The world was spotless of the virus that came in and quickly impacted the world. How did this small virus change the world as we know it? What made it so dangerous in such a short time span? To really answer this question, we need to go back and study the genome, the virus's blueprints and components. So far, we know two types of cold viruses, the rhinovirus and the coronavirus. As you've probably already guessed, COVID-19 is a part of the coronavirus family, and with that comes specific advantages that make it superior in infectivity. Starting with their own similarity, both viruses have a positive sense RNA and a protein capsid. Fun fact, the positive sense allows for the virus to directly translate information in the cell, rather than having to create a new strand for the cell to translate during replication. The key difference, however, lies in the fact that coronaviruses contain an envelope around their capsid, while rhinoviruses lack this structure. Envelope viruses are able to more easily travel and spread through body secretions, such as blood, water droplets, sneezing, etc., as well as not killing the host cell once done with replication. However, they are sensitive to drying and heating. This flaw is minuscule when we see how long and how much the virus can persist when enveloped. This is great and all, but how does COVID-19 become better than the members of Corona? Great question. The answer to that lies in the name Corona, or crown. See, each coronavirus has a rim of spikes covering the outside of it in a manner similar to a crown. These spikes are surface proteins that contain a receptor binding domain and fusion region. These are responsible for fusing and communicating with the host cell. They are also the COVID-19's greatest weapon. Each coronavirus has a different type of spike in terms of shape, structure, and function. Example is the bat COVID and SARS, close cousins of COVID. In the bat spike, you see two cleaves. In the SARS, you will see one conjoined spike. As for COVID, you have two sections and a protease cleavage site containing a furin recognition. Furin is an enzyme that stops or allows other proteins and amino acids to function during the mature or active stages. This is essential in order for the virus to quickly enter and evade immune responses. Keep this in mind for later. When looking at corona functions, part of the genomes also contain key differences. Example is the virus's coded affinity for ACE2 cells. ACE2 cells are cell membranes containing angiotensin-converting enzyme. These are found in the lungs, arteries, heart, kidney, and intestines. So what does this all mean? Well, we know COVID has the ability to spread in many forms and persist in cells for a long while. We also know that COVID is able to use their specific spike protein in order to interact with ACE2 cells, creating an effective strategy to target a majority of the body. Finally, its furin recognition site allows for the virus to activate the enzyme and allow for it to better enter and fuse itself into the host cell. These traits allowed for COVID-19 to become an effective virus that was easily and quickly able to spread. But there is still hope. By knowing how the virus is successful, treatments and vaccines can be used to turn its strengths into the virus's weakness.